In a laboratory nestled in Beijing, a perplexing scientific enigma resides. It is a human DNA sample whose very existence seems to defy the established understanding of early human migration into Asia. It was meticulously extracted from skeletal remains discovered deep within a limestone cave system in southwestern China. Subsequent radiocarbon dating placed its age at approximately 34,000 years old. The crucial sequencing of this ancient DNA took place in 2019, leveraging the most advanced genetic analysis technologies available. Scientists expected another piece of the puzzle, and the results emerged. They presented an astonishing, undeniable mystery. Researchers rigorously checked and rechecked their findings, running the analysis multiple times, fearing equipment failure or contamination. But each confirmation only deepened the mystery. The genetic markers, the unique fingerprint within this ancient genome, simply did not align. There was no match. Not with contemporary Chinese populations, not with ancient groups previously identified from the region, not even with populations from Southeast Asia, Oceania, or Central Asia. In essence, this genetic signature was completely alien to every established migratory path and settlement pattern of early humans across the continent. This DNA represented something profoundly different. It was a genetic blueprint that steadfastly refused to integrate into the accepted narrative of how humanity dispersed throughout Asia. It was as if this individual belonged to a ghost lineage, a branch of humanity that walked the earth and then simply vanished, leaving no descendants behind. To truly grasp the extraordinary nature of this DNA, it's essential to first understand the typical process and expected outcomes of ancient DNA analysis, and why a zero-match result is almost statistically impossible. When scientists extract and sequence DNA from ancient human remains, their primary method involves comparing these new genetic sequences against extensive reference databases. These invaluable databases are meticulously populated with genetic information derived from both modern populations worldwide and a growing collection of other ancient human samples. Such comparative analyses serve as powerful tools illuminating the intricate tapestry of human ancestry. They are capable of revealing the direct lineage between ancient groups and their modern descendants, charting complex migration routes and documenting the nuanced ways in which populations intermixed over millennia. This sophisticated system operates effectively because, for the most part, human migration followed discernibly predictable patterns. Ancestral groups embarked on their journeys from Africa, venturing into the Middle East, subsequently fanning out into Europe, Central Asia, and South Asia, and eventually reaching East Asia. The genetic trail left by these migrations is remarkably clear and consistent in the vast majority of cases. However, the Chinese sample, with its confounding genetic profile, emphatically disrupts this well-understood pattern. This ancient DNA exhibits markers that are conspicuously absent from any other recorded genetic data. We are not talking about mere trace amounts that might hint at minor intermixing with an uncatalogued group. Rather, substantial portions of this ancient individual's genome possess no discernible match in any existing database. It presents a terrifying scenario for anthropologists, as if this particular population diverged from all other human groups at a very early stage and subsequently evolved in utter isolation for thousands of generations, only to vanish without leaving any genetic legacy in modern populations we recognize today. While such a trajectory is theoretically conceivable, it is considered exceptionally improbable. Human populations throughout history have been characterized by constant interaction and intermingling. Migration, trade networks, and even conflicts have consistently created ample opportunities for genetic exchange and admixture. The notion of a human population maintaining complete isolation for tens of thousands of years, especially when surrounded by other human groups, fundamentally contradicts the standard model of human demographic history. Furthermore, the geographical location of this find makes this isolation even more baffling. Southwestern China during the late Paleolithic era was far from an isolated wasteland. It was a thriving hub, Extensive archaeological evidence unequivocally demonstrates a vibrant landscape of human activity across the entire region during this exact period. Discoveries in the region include sophisticated stone tools, striking cave paintings, clear evidence of controlled fire use, and meticulous food processing techniques. These findings collectively paint a picture of multiple human groups coexisting, often in relatively close proximity. The profound genetic isolation implied by this DNA sample thus forces researchers to confront a difficult dilemma. Either this population somehow managed to entirely avoid all contact with its neighboring groups, a notion that strains credulity, or there is another, as yet unknown phenomenon at play that current scientific models simply fail to account for. In the spirit of rigorous scientific inquiry, the researchers published their findings with commendable caution, proposing several potential explanations to account for this massive anomaly. Perhaps, researchers suggested, this sample represents a previously undocumented wave of migration into Asia, one that arrived even before the primary migration. Alternatively, they considered the possibility that this population was geographically isolated by natural barriers that are no longer evident in the current topography. But neither holds up. If an earlier wave of humans reached southwestern China, we should expect to find their genetic legacy somewhere. Human populations simply do not vanish without leaving any trace. Genetic mixing almost invariably occurs, much like Neanderthal DNA in Europeans or Denisovan DNA in Oceanians. The geographic isolation theory also fails. Southwestern China in the late Paleolithic was an integral part of a vast continental system. For complete isolation, it would require barriers so extreme the population couldn't leave. Yet, paradoxically, other populations could somehow deposit stone tools and cultural artifacts right next door. The data quality theory, focusing on issues with severe DNA degradation or modern contamination, is arguably the most compelling defense against this mystery. Ancient DNA is inherently fragile. As molecular bonds, the molecular bonds slowly shatter, breaking the genetic code down into smaller and smaller fragments. It is a well-documented scientific fact that DNA extracted from hot, humid tropical and subtropical regions, much like southwestern China, degrades at a significantly faster rate than DNA that has been safely preserved in the freezing, stable conditions of Arctic climates. So, anthropologists asked a vital question. Could this natural degradation process specifically create the false appearance of completely unique genetic markers? Could the decay mimic a new species? For degradation to generate such a specific outcome, the patterns of decay would have to be extraordinarily precise. It would be like a book rotting away, and the remaining letters perfectly forming a brand new, coherent language. It would mean naturally creating genetic markers that look exactly like real, functional human DNA under a microscope, but conveniently fail to match any known human population in the entire global reference database. In reality, random genetic degradation typically renders ancient DNA completely unreadable, leaving behind obvious, jagged errors in digital artifacts. Random decay does not miraculously invent brand new, coherent, and perfectly structured human genomes out of thin air. To completely eliminate this doubt, the Beijing researchers thoroughly examined the specific molecular degradation patterns across the entire DNA sample. They meticulously mapped the decay, and they found that the highly unusual, alien markers predominantly appeared in the specific regions of the ancient genome that demonstrated absolutely excellent, pristine levels of molecular preservation. Any areas of the sequence exhibiting obvious signs of degradation were scrupulously excluded from their analysis. This anomaly is undeniably real. This leads to the most compelling possibility. What if our standard model of human migration into Asia is not entirely wrong, but rather drastically incomplete? It might be missing entire populations in migration waves that have simply not been documented because their genetic legacy is exceedingly small, or we haven't unearthed the crucial samples. We have scattered data points separated by thousands of miles in millennia. Entire populations could have existed within these undocumented intervals. The specific timing of this sample, 34,000 years ago, places it within a period of profound global climate change. The last glacial maximum was approaching. Populations that had thrived might have been completely displaced or entirely eliminated by radical environmental shifts. This sample could represent a doomed population caught in the throes of this massive transition. If this sample genuinely represents a distinct, previously unknown population, it unequivocally means that our current understanding of ancient Asia is missing critical pieces. Entire populations that thrived and disappeared. This wouldn't be unprecedented. The Denisovans were utterly unknown until DNA was sequenced from a single finger bone in Siberia. The southwestern China sample might be pointing towards similar revelations concerning population diversity within modern humans themselves. The Beijing researchers acknowledge that additional samples are absolutely essential. They have issued a call for renewed archaeological efforts. Finding more samples could take decades or might never happen. For now, this enigmatic genome persists as a profound anomaly, a ghost in the data, continually reminding us that the story of human origins is far from finished.